mode. Good day to everyone joining us and welcome to today's Excellience webinar featuring Cambridge Major Laboratories, Integration of Solid-State Chemistry and Process Development. My name is Donna Papacosta and I'll be your host for today. This seminar will run for about 45 minutes, including a Q&A session toward the end. This webinar is designed to be interactive, so please feel free to type in questions and comments throughout the presentation by using the questions function on the panel on the right-hand side of your screen. If we cannot answer your question during the allotted time period, we will send you a direct reply after the live webinar. We will also be running a few polling questions. We hope that this will drive audience participation. We appreciate your participation and we will share the results with you. At this time, all participants are in listen-only mode. This event will be recorded and made available for future download from the scientific seminar page on the Excellience website. The presentation slides will advance automatically. If you need technical assistance, please contact GoToWebinar at the numbers displayed. In the U.S., 800-263-6317 or 1-805-617-7000. Now we'd like to begin the formal presentation. Cambridge Major Laboratories is a leading global chemistry outsourcing partner to the pharmaceutical and biotechnology industries. CML produces pharmaceutical intermediates and active pharmaceutical ingredients from early preclinical development to commercial manufacture. CML boasts an impeccable reputation for delivering some of the most challenging projects while meeting the tightest timelines. Cambridge Major's reach is truly global, operating 3 facilities in the U.S. and Europe. CML is organized by 5 key integrated centers of excellence: process chemistry, API manufacturing, analytical services, solid-state chemistry, and quality assurance. With a full suite of services and strong industry partnerships, CML can serve all of your drug development needs. I'd now like to welcome our speaker for today's seminar, Dr. Edwin Arret. Edwin is a solid-state scientist at CML. With the background in physical chemistry and over 20 years of experience in growing single crystals of small organic molecules, he now leads the solid-state group at CML Europe. Next to crystallization expertise, also form selection, polymorph, and morphology studies are implemented in the drug development programs. Before joining CML, Edwin worked as a project manager in the high-throughput CRO Avantium Pharma and extensively studied the polymorphic behavior of organic dyes for the photographic industry. Edwin has several inventor patents of near-commercial APIs and often publishes and presents his applied approach of solid-state chemistry in drug development. And now, without further ado, I'd like to hand the microphone over to Edwin Arret. Edwin, you may begin when ready.
Thank you, Donna. Yes, today we're going to talk about integration of solid-state chemistry and process development. Solid-state chemistry is a very broad field and also called life sciences, and most of the things we talk about are crystallization, applied crystallization, and now in combination of a process development. I'm a solid-state chemist, so there will be no organic synthesis routes here. Mainly the integration of the two and the benefit of the two separate when it's combined. No, it's not working. I'm sorry, but my presentation is not working at the moment.
Edwin, yeah, I think you did just the right thing. Just try to show your entire screen and then try to advance. Yep.
This should be okay. It was locked. I'm sorry. Okay, outline for today. A little bit about CML. I will talk about drug development, solid-state chemistry, why integration of the process development and the solid-state chemistry. We talk about the 2 approaches that we have: Form Select and ProCryst, and 2 case studies. As told, CML is a global leader in API development and drug substance manufacturing. The focus is the chemistry. So we are CRO for pharmaceutical and biotechnical industries, and chemistry is the main topic that we have. By cooperation with other companies, we have designed a virtual one-stop shop for all other services. It is not included in the company itself, but we use other companies to cooperate with, and they have their own expertise. And together, you can benefit of the knowledge of all. As said, we have facilities in the US and one facility in Europe. I'm located in Europe at this moment, and this is the main R&D of solid state what we have here. Cooperation is also with the US, so projects that run in the US also benefit from the solid state R&D which is done in Europe. Where are we located in drug development? Mainly after discovery and a little bit of optimization, we take candidate nomination, and we go from production of gram scale up to, in Europe, up to kilogram scale, tens of kilograms, but in the US we can go further, even up to commercial production of APIs. And we serve the whole field, what is needed there. So drug development. Of course, R&D is everything where it starts with. You have to find out what is going on, what is possible, but we work according to the ICH guidelines for all GMP studies that we do. The final product will be produced under GMP if needed. If you only need a part of the whole process, that is also possible. If only, let's say, scouting of what is possible of your process, that is also one of the things that we do. Next to that, we deliver a lot of different things there which are additional to the product itself, the API itself. So if needed, we can deliver the CMC section, we can deliver input for a patent, and anything that is needed to develop your compound that can be added by cooperation with other companies, or just select the things that is needed for your compound. One thing that is central here is quality and reliability, and we see the work that we do is a part of the total picture of drug development. So what we need to do is connect to Medchem and sometimes high throughput companies that screen already, use that information, take it into what we do in development, and hand it over to other companies that are working in formulation, in tox studies, but also in larger scale production. So we treat the compound as it is our own, and we make sure that everything that we discover here will be handed over in the right form to the right people, and make sure nothing gets lost on track there. The end game is finally to have a clinical trial there. Main thing in drug development is to make choices. Which compound, which API do we take forward? If you want to make such a decision there, what you need to know is information. As much information as possible at the right time, but not only information to know, to show what you can get out of a small amount of compound, but just know what is needed at the right time. So know if it's chemically possible to make the API on a larger scale. See if the physical form of your product is okay, if you don't have stability issues, if you don't have any issues on low melting point or anything like that. See if the stability is okay. Check if you have the intended formulation, if it fits with the properties of your compound, and also have a good risk management to go into further scale up. Know what you're getting into, but also know what can be on the route further when you try to scale up your compound. Main part, what I'm doing here is solid state chemistry. So why? The aim of this solid state chemistry is to get a solid form which you can get each time, a reproducible properties like your solubility, dissolution rates, bioavailability, but also have a look at your compound itself, that each time when you produce it, you have the same form, you have the same particles, the same particle size, and connecting to that the same properties of your compound. You need to find the suitable form for that stage of your development. So if it is only a selection of different molecules and you need to take one of these compounds forward to see which one can be produced easily, then there is less needed to know than when you have one compound and one only, and you want to take that as far as possible. Find out if you need a salt or a co-crystal even. Find out what is the polymorphic behavior of that form, but also have a look at the crystal habit of your polymorph. Most of the properties of your producing of your compound are related to crystal habit. So if you have poor quality material, still the polymorphic form can be okay, your salt can be okay, but you have problems with crystal habit of your form that you have selected, and that can also be solved. We approach it with a scientific background of the experiments. We use, if needed, DOE or quality by design. If needed, that's the emphasis there. If it is only to know a little bit of it, we do a full factorial of what is needed in that part. But also, even important is the theory that we use is only to explain what is going on, and we only use applicable conditions to produce the compound. It is not that because in theory it should be doing X, we're going to look around to that point, even if it is using methods or solvents which you will never use in further development. So indeed it's a background, but it is not the leading part here. So only pharmaceutically acceptable conditions we're going to use, and then we already look to commercial manufacturing. So that is the use of counter ions, the use of solvents, but also the use of conditions. Main thing is to be selective and conclusive. Why? You need to make a decision to take one forward, and it cannot be that yes, maybe in the future you can work with this. It is yes or no, and this form is best to take forward now. That can be that we advise to look for a different form if your material is absolutely not stable under normal conditions. Especially the solid state chemistry. Why? Well, we talk about crystallization here. That is the main part, and it is an effective way to purify your product. So if you have any impurities during the process or in your final product, it is the best way to scale up a purification process. But also have a look when to purify, so when you should have this crystallization step. It can be at an intermediate step, or it can be at the end if you collect everything and simply have one simple step to remove all these impurities. Second is selection of your optimal form at the earlier stage. Why? Find out which form you can use. So it can be that it is the most stable form, but it can also be the most stable form under these conditions. If you have a perfectly stable anhydrous form, but as soon as the conditions are such that you raise about, let's say, 10% humidity and it turns into a hydrate, we do not advise to go for this anhydrous form. You go to a hydrate because that is stable under the most conditions, the most storage conditions, or when you use it for. Optimal form means the one that is easiest to form, to make in your process, but also the one that you can handle, that you can store, and that you can use and you can reproduce each time. So no batch-to-batch differences, no transformations, and also no stability issues. Finally, crystallization conditions determine the quality of your product. If you have a good crystalline product, you have good defined properties. As soon as you go into mixtures or partly amorphous, you will lose part of your quality of your product. Part of stability it can be, part of properties like dissolution rate or anything like that. So improve your crystallinity and improve your particle morphology there. When should we have solid state? Well, the part that we are doing now is from lead optimization and going into clinical development phase 1, phase 2A. So during your process development, to go from R&D to GMP material, it is best to have already a selection of your form, do a polymer study, look at the crystal habit, and before you make your GMP material, know which form you will take forward, know which forms are out there, and which are the critical factors in your process. What do you need to know of your process in further on, or what do you need to investigate more in depth when you have more material available and when you have shown that your compound, your API, is really the one to take forward. A small overview, what do we do? We have a look at material crystallization and characterization. I'll come back to that later. But also solubility determination. That can be in organic solvents. It can also be in different buffer ranges. It can be in simulated fluids, depending on what will be the purpose of where do you want to know the solubility of. Salt selection or co-crystal selection are the things that we use here, but also polymer studies, not the very broad polymer screens of hundreds or thousands of experiments, but we look in depth to the conditions that may occur during your process. Crystallization process development, the crystallization of a compound and the scale-up of that specific form is one of the most difficult things to do. So a program to crystallization process development is also needed before you go from a few milligrams up to kilogram scale. Finally, we also have a look at crystal habit optimization, and that means that we have a look at particle size, particle shape, but also roughness of your crystals if needed. If that is giving the wrong properties, we can have a look at that and see how we can optimize these. As told, I will go more in depth into crystallization. Here's an example of a crystal, the block shaped in the picture here, and there are some small needles around it. When you look at it chemically, they are totally identical. When you look at the polymorphism, so an X-ray of this one, they are totally identical. But it's just the conditions under which you grow these crystals that makes it that these two forms may occur. So if you do not control your conditions in a correct manner, then you can get a combination of these two, and you have a very broad particle size distribution. And connected to that, of course, also the properties of your material are changing. That means that solubility or dissolution rate can change here. So why do we do crystallizations? Well, one is isolation or purification. We talked about that one. But it is also crystallization of a specific polymorph. So when you know which one you need to have with the right conditions, you also have a look around that polymorph. What is the area in which I can do? What is the temperature range? What are the different solvents that I can use for this specific polymorph? And that you don't get any side effects of amorphous content or other polymers which can be there. We look into crystallization of oily or waxy solids, which most of the time it is caused by a very high solubility of your compound. It refuses to nucleate, and crystallization does not occur. You get a semi-solid or even an oily material. These are impossible to handle when you look at normal conditions of producing. So you keep them in solution and you take everything with it, all the impurities and everything you don't want to have, you take it with it to the next step where it might influence your process over there. So if you can have a crystallization of your oil, you remove again impurities, you have a solid, you have a stop if needed, and you can improve your process and the quality of your product there. Crystallization of amorphous, so that is when your material crashes out of solution. Most of these processes result in crash crystallization because the group is added and all of a sudden it does not dissolve in your material, in your solvent at all. So the material that you get does not have the time to grow as crystals and crashes out as amorphous. Crystallization conditions, most of the time it is a more controlled cooling or more controlled addition of an anti-solvent may result in a crystalline product instead of the amorphous one. But we also specialize in the materials that simply grow as amorphous and need a little bit help to crystallize. So a co-crystallization can be there. It can be using solvent, so that is making a solvate there that is only useful if the solvent is acceptable and not ending up in your final API. Co-crystallization can also be used by other solids, so use something to make sure that you have a crystalline lattice there to avoid amorphous or to avoid an oil. Co-crystallization means that you have to look at the molecular level to see which connections are possible, how to produce an acceptable co-crystal, but also guide it to an acceptable API. There are two other things still there. That is, on the other hand, if you want to stabilize your amorphous, so if your solubility of the crystalline form is that low that you cannot use it as an API, you need to have other options there. Stabilizing amorphous in a polymer lattice can be one of them. But also avoid the crystallization of unwanted polymers. If a certain sulfate or a certain other polymorph is available and you don't want to form it, either because of a patent or because it influences the yield or the properties of your polymorph, we can have a look at how to avoid these polymorphs, but also find a way how to control it and to see that you don't have that polymorph during your crystallization process. What are the methods that you can use? Well, this is a full screen with totally to the left the research conditions. These are mild conditions, they are close to equilibrium, and they can take up to months. In production, well, these are seconds to minutes because conditions are severe and these are totally non-equilibrium. Can we use all of them? Sure. If you have the time to use them, if you want to use them, of course. Can we scale them up? No. For that, we only have a few limited crystallization methods, which mostly are cooling and anti-solvent. Sometimes when you have it in the reaction, you have a reactive crystallization, you cannot avoid it, but you can have an additional recrystallization going on if it is your final product and you need to optimize it. Finally, also some slurrying can be there if you have not the correct form, not the most stable form that you want to have. You can slurry in certain solvents to make sure that you have a homogeneous compound there. So the main thing is only to use scalable methods and not all methods available, because you're not going to use them when you go into production. Cooling crystallization is one of them. What you have here, you start with point A, you have a solution, you cool down and you keep the concentration in solution that stays stable. So you end up with B, which is the point at the solubility curve. At that point there's equilibrium. If you have endless time and you wait at that point, you will get crystals. Most of the time you do not have that, so you cool down further to the form C, where you crosses the nucleation curve. C means solids are formed, your concentration drops down, and you go back to D, which is most of the time around room temperature, and that is the amount of solid that you can get out. Why do we present this? Well, that's the next slide. What you see is when you cool down, that your solubility curve, that one is fixed. You have your material in there and you have your solvent there. That one is something that is also described by most patents. If you have your process fixed already, that means that you cannot change anything there, but that is not depending which form you will get. Nucleation curve, that one is that can be varied, because it is determined by crystallization conditions. That is cooling speed, but it is also what kind of material do you use, what kind of impurity level do you have, do you use seeding or not, because then you don't have nucleation at all, but you have just crystallization growth. And that nucleation curve, that one determines which form will be there, which form is accessible. The different forms and different conditions may lead to different polymorphs that you get, but it may also lead to batch-to-batch differences. So to control your crystallization, you need to know what are the possibilities to influence your nucleation curve, how broad is the metastable zone width, that is the area between the solubility curve and your nucleation curve, and what can you do to get the right form each time, each batch. For anti-solvent addition, you can have the same story here. There is a solubility curve which is fixed, and there is here a metastable zone limit, which depends on how much anti-solvent do you add, how fast do you add it, how much time do you allow to have it there, what is the temperature which you use. So that is the same things can occur there. If you do not control the addition of your anti-solvent, you do not have batch-to-batch same form and the same quality material. How do we work here? We determine metastable zone width because it allows you to have an idea how well will this compound crystallize. You determine the solubility in several process solvents in several temperatures to have an idea how well does it dissolve. In these cases, the solubility curve is as noted here in this figure, but they can also be more flat or even totally different when depending on the temperature. Up to now, I only have seen this kind of behavior, so we use this one, and you can use cooling crystallization for most of the compounds that we see here. When you measure your dissolution and nucleation temperature at different concentrations, you can have an idea how broad your metastable zone width is and what is the area to play with. If you do it also applying different conditions, so different cooling rates, you get an idea on the different forms that can be reached by this method, or that you have to go to more extreme conditions, which is anti-solvent addition or crystallization. Of course, when you do something like this, you dissolve everything, you get a complete solution, you cool down again, and you get a form. You need to check by X-ray or other means to see if the form that you make is indeed on each temperature the same form. So it cannot be that you just measure this and you know everything. No, you also have to check if you still have the same form, if the craft that you're making there is indeed of that polymorph and not of something, a solvate or anything else that can be there. Next is crystallization process conditions. When you have defined the area in which you can crystallize, you can nucleate your compound, and you have defined already which solvents to use. Here we use the Multimax equipment for the first scale-up. In general, we do steps of approximately 20 times up to 50 times if the process is really going well. You can have a look at the cooling rate if it is still scalable, if applicable the seeding conditions, anti-solvent additions, and optimize these crystallization conditions. At this scale, we may use design of experiments, but this is still on a small scale a parallel screening of what are the conditions to check the optimized crystallization conditions there. When we go to even larger scale, so these are really the process conditions which we have here, it is 1 liter most of the time. You go from a parallel experimentation to single experiments, and from that stage onward you actually producing different batches one at a time. So you have a demo run, you go up to kilogram, you go to the pilot plant. These are the steps that we make, and all the information that is obtained in R&D and in the parallel experiments can be used at this scale. One thing is most of the time not usable, and that is when you say I have to wait for 3 hours at 50 degrees before nucleation occurs. Most of the time when you look at things like time to crystallize, it is not scalable at all, and you need other techniques to check if indeed your nucleation occurred, if indeed at that scale crystallization is at the same stage as before. But all the information that you get can be obtained from the small-scale R&D experiments. So in summary, solid-state chemistry, we have scalable production methods, cooling and anti-solvent, acceptable process solvents, class 3 or 2, or easy to reduce to allow to residuals level, and material properties, a stable form, and good filtration and further handling of the compound itself. These are the things that we can use and to select one form to take forward and to combine with the process and really have an integration there. Why should you have an integration? Well, the timelines, these are shortening each time, and parallel development of both your solid-state chemistry, your process development, that saves time. I will show a small part in the next slide. The early integration of solid-state chemistry and process chemistry gives a chance to have a good risk management of your drug development. You have already an idea what is possible, what are the dangers that you get when you scale up. Are you going to see multiple polymers? Problems with isolation, filtration can be very slow. These are things that may occur when you scale up and when you go further in your drug development. So as soon as you know that it may occur, you can have an idea on how to approach that problem at the earliest possible stage. When you couple both, you have already the idea of the problems, but also your result will be a better production process and a better quality material at an earlier stage. You cannot optimize a process and have crappy material at the end. You need to have a good material because that means that you had a good process. You thought about as well the chemistry as well as the physical form of your compound. Very short timelines. Well, it's clear that when you integrate solid state, then you can do it in parallel, and some things are not needed anymore. Handover of any information is much easier than when you have two different companies who work on this one. Characterization of the material is not needed because that is done already while the process development is ongoing. As soon as in the process a problem arises, you can step in and say, okay, this is needed to get the process going again. And that can be after a few weeks, it can be needed from the start. So you can save up to 3 months of development time here when you have this integrated approach. But most of the time it can take up to several months, up to a year, which delay you will have, because you go from process development, it doesn't work, you first have some solid state going on, back to process development, have to repeat certain things, not optimal, and so on. That is something that really, well, it is to avoid, and by the integrated approach, you can indeed avoid that one. So we have two approaches at CML here. When a process is already optimized or when it is fixed, we only have a look at the form. So form select means that most of the time the HCl salt or the free form is taken as default. So you can make an HCl salt out of it. Okay, let's take this one. We go forward with it because we always do that. Many of these compounds, they are not optimal, because also HCl salts have the tendency to be hygroscopic or to form hydrates, and that can be uncontrolled hydrates, so you do not have a good idea what actually the active ingredient is when you have a mass of API there. So it is not an optimal situation when you just take each time the HCl salt or the free form. The choice of solvents, the choice of salt and the choice of solvent from which you get it greatly improves your properties, and that can be solubility or stability. When you look at the earliest possible development phase to this form, you can have an effect on the crystallization and to see which forms or which salts can be selected to study in parallel, and then make a selection, because the aim is to compare these properties and select the most optimal form. The process is already fixed. Details are there if you make a salt out of it or you have a recrystallization going on. I will show a typical program, which is when you have a look at a possibility to form any salts. It is about 12 to 24 counterions and the free molecule itself. 2 to 4 solvents, including water and methanol. Water is very important here because everything turns on the aqueous solubility. That is the main point here. If you want to make a tablet, it has to be soluble in water. Otherwise, you will have huge problems to get it to be active at the right side there. Dissolution rate comes at a later stage, but it is as equally important, of course. When you have a look at which methods to use, well, 1 or 2 methods. If it is a very insoluble compound, you just go to slurry at different temperatures. When you have a choice of different solvents which you can use, then you have cooling crystallization, which most of the time improves your quality of your salt. The ranking criteria, because you are not putting absolute values there, you are looking at which one is best to take forward. It's very much depending on what do you want to do with it. It depends on which property you want to improve. So you can have a look at crystallinity, you can have a look at aqueous solubility, or you can have a look at stability. All of the ranking criteria needs to be looked at before you start the program, because if you have a problem with your solubility, the choice of your counterions will be such that in general these counterions will improve the solubility. And when you know it doesn't want to crystallize, you go to larger counterions or find a way to influence your nucleation behavior and to crystallize it in a better way. The second approach that we have is when next to the choice of your compound, of the form of the compound, you also have to develop a process. So when you have a problem with the yield that you get, or that you don't know exactly when the end is of your reaction which is going there, you have possibly something which is well, very difficult to filtrate, and impurities of each batch are different, or you just have a poor quality material. So all these things may occur in the process, and when you look only at the chemistry and you try to solve it, of course you can improve, but maybe you cannot solve the real problem which is behind this. And that can be that either your crystallization is not okay, your form is not consistent each time, you have mixtures, you have a lot of amorphous material available. These are all things that may occur. So that is why we combine process chemistry and crystallization science, and by working together in this process, on each and every step you can improve both. You can improve the chemistry and you can improve the solid state. So the aim is smoothen this R&D route to optimize the end product, but also have a look at what is the process time, what is the yield, other things that are important, which depending on what do you need to improve there. I have 4 examples here on how to do it. You can have a look at the yield. So by looking at your metastable zone with again, you can calculate how much will be present at high temperature, how much will be present at your low temperature, room temperature, or anything. And the difference is how much yield can you get. Maybe you have to select a different solvent if your nucleation curve is very flat and you cannot get a lot of material out of your solvent by just cooling down. Your cooling conditions can be very important. How far to cool down, but also how fast. Seeding can be important. Sometimes you just get a little bit of hazy solution and it doesn't really want to crystallize. You go down far below zero and finally it crashes out and you have a poor quality material. If you seed, you can control it better. Control means that you can get a good material out. Maybe an anti-solvent needs to be added. If a lot of material still remains in your solvent in solution, then anti-solvent may help you there. Filtration, of course, each time, each process has some of these bottlenecks where you have poor filtration, where you have a very needle-shaped morphology of your crystals, and these block your filters completely. So you have to wait for days before something is really filtrated or before everything is dry. So the conditions on which filter to use, pressure, temperature, that can already have an influence, but also which solvent to use for the filtration, for the isolation of your material, and which solvent to use to dry it, to help it dry. Maybe anti-solvent can help there and even wash away all the impurities that you have. But mainly control of the particle habit, and that means the shape and the size. That one is very important in filtration, to improve your filtration rate and to improve your drying properties. The stability of your particle is one of the issues there. Purification. Of course, recrystallization is easily added. You have a process and in the end you don't have a pure product. Well, add another step in recrystallization. But you can also think of remove this impurity at the moment that it is produced, and if it cannot be isolated from your product at that moment, look at different forms, look at intermediate salts, look at possible solvates. For an intermediate, it is not important what the form actually is. If it is most stable for years, no, you just want to have a purification step and immediately go on with the process. So select the process step in which you can remove these impurities as easy as possible. And the final product, there it's different because the API has to have as less residual solvent as possible, as less as impurities as possible, and to have a homogeneous particle size distribution as much as possible, of course. So when you selectively and controlled crystallize the form, you can have a better improved product. These are separate steps, and the process itself determines which steps are needed. I have two case studies. One very easy, select a salt. It's just to go into phase 1 studies. There are two things that are important: long-term storage as a dry solid and a fast dissolution in water. These were the two things that were actually that needed to be improved. Materials were available, and they had an amorphous HCl salt with by HPLC 80% purity. The freebase which was made from this HCl salt was even worse. We can do a lot of to purify the product, but there is a limit, and the amorphous freebase was no good starting point for this kind of studies. So the approach was such that we isolated freebase from the HCl salt, and by a more controlled isolation, the use of different solvents, we had to look at what solvents to use. What we had was again an amorphous freebase, now improved purity, and there were no residual solvents present. So that was the first step to get a good starting material. 75% purity is still very low, and you have a lot of impurities there, so this is again not ideal to have a salt screen going on. So the next step, find a way to crystallize it. A small polymer study, in fact, or a small crystallization study to use 24 different solvents, and in the end what we had was a major improved purification step and a nice crystalline product of it. With this knowledge, we went back to the process and we implemented this purification step before the final step was done, because in the final step we could not combine the need what was for the process itself and a crystallization. So we go back one step, and there it could be implemented. There the main impurities were already present, and the purification was very useful at that moment. When you go to the crystallization solvent there, that one was also used as the final synthesis step of the freebase itself. After that, immediately the HCl of the salt was prepared, so it could not be implemented as a purification there. Salt selection, because now we have a freebase which is nicely pure. 15 different counter ions could be used to improve the aqueous solubility. So we had already the first step that we have a stable form, but now the aqueous solubility was the main thing to focus on. We had two different solvents to produce. What we saw that some weak diacid, they bridge, they made solid material, not that very crystalline, and it shows poor solubility, even worse than the freebase itself. But the very strong acids, and that in fact these were all but the HCl salt, showed nice crystalline material and an improved solubility there. So to see which of these strong acids to use, again a polymorph study was done. Everything was done in parallel here, and to find a good stable non-hygroscopic salt form. From that we checked which form we could use, and we went to TMP kilogram batch production, and in fact the process development itself took longer than the selection of the right form. So the form selection was no longer on the critical pathway of that development. We saw some problems with the starting material, and while doing these steps of producing, we also had an idea of how to select and which form to take. So you do not lose any time when doing it in parallel. Last case study is 8-step synthesis, but in this case the process itself was already a little bit fixed, but the final salt and the final conditions, the impurity levels, everything was already fixed of the final product. There were already tox studies done on this compound and they didn't like to repeat it. Of course, you need to avoid this kind of situation, but in this case we had to work with the result is already done. What were the issues? No crystalline intermediates, no scalable purification was available, and also the final step was not in a very acceptable solvent. So you have to dry a lot before you can even have an acceptable API at the end. This means that the whole process took a very long time, and still the purification, well, you don't want to have a column in it, but it was needed in this case. So the approach was that each step, each of the 8 steps, we had a look. First, synthesis. Second, solid state, and have a combined opinion on how to approach this step, how to optimize this step. First of all, there was a purification step. We had an intermediate oil, and what I stated before, we looked at the possible salts, so we defined a crystalline salt, and the intermediate could be isolated there. Of course, you introduce an additional step there. The overall yield was 3 to 5% lower, but now you have a purity of 98% plus each time and consistently. And you have a material which you can isolate, which you can say, this is our new step. You can see if everything, you have a better control on the process. So also the isolation of this was very fast filtration, so not really anything wrong with this purification step. It was just add counter ion that could be done without any other changes to the process itself, and instead of isolation of an oil you get a crystalline salt. Another step was, well, it was an extreme slow filtration. It formed a thick cake of very fine particles sticking together, and as soon as the solvent was gone from the surface, water was attracted. So in fact what you needed to do is a filtration with nitrogen flow on top of it, very controlled conditions. As soon as you slow down that nitrogen flow or remove it, you try to handle your compound, it liquidized immediately. So we use several common solvents there which were suitable for this step. First of all, you have a look at what is the chemistry, what can we use, and these were used to see if anything was done. So there were several solvents, as long as it was not an alcohol and you have a fine white product. When you use acetone, it even gave block-shaped crystals, and that gave a very fast filtration, but also no residual solvents. And again, within 1 to 2 weeks, we had the next step looked at. The chemistry was okay, but also the form that you get afterwards was okay. Finally, the final product, the salt formation itself, well, it was produced from dioxane, and that was actually not possible to do a good crystallization from. So we had a look at which you could use when you have a dry alcohol in the process. Then you can steer still on yield and the purity of your final product. So what the message in fact is that when you connect both the process development and the solid state chemistry, it shortens the timelines, but it gives a better process, and it also gives a better form. You don't need to repeat anything of your development because you have chosen the wrong form or you are stuck in your process when you start to scale up. What is also important is to fix the delivery method as early as possible. If you have an idea how to get it in at the place that you want to have it, that is very much depending what characteristics of your API you need. What do you need to improve? Of course, a tablet is different than a topical, and everything has its own optimization route, and it also has its own solid state part which can be applied to this compound. When you have possible issues in production, like a purification method which cannot be scaled, look at them, recognize them, and have an idea how to optimize it. When you control your crystallization, you already have, well, let's say, half of the work done, because you know what are the factors to improve your crystal size or your crystal shape. And finally, do not collect all information that you can, because it is possible, but collect these data sets that is needed to know and focus there on the appropriate what is needed to know. You can have a lot of data points and still not have the overview, but only look at what is necessary to do at the early phase. This was the approach of the combination of solid state and process development here at CML. And if there are any questions, please.
Thank you very much, Edwin. Yes, we're now moving into the question and answer part of our program, and we will start with our polling questions. And the first polling question, and you'll have a few seconds to answer this, what is the most important to you when you are outsourcing a solid state service provider? Speed, expertise, price, quality, or integration? So please take a moment to answer that. What's most important to you when you're outsourcing solid state services? And you can just click on submit and we will see what those answers are. So Edwin, we have 60% saying that expertise is most important and 40% saying that quality is most important. So no one chose speed, price, or integration. So do you have a comment on that?
Yeah, of course, expertise is indeed the thing to go for there. Each has its own field, which it is, of course, best in. And when you go for the best because you want to have value for your money. And quality, second, of course. You want to have the best product in the end. So the other options that were there are mainly well, benefits that come with the choice that you do. When you select the approach that we do, indeed speed is one of the things that is added there. But we do not lose the quality or have less expertise in this field by let the solid state do by, let's say, organic chemist. No, that's not the way to go, of course.
Right, okay. And our second polling question, with assuming timelines are the same, does proximity or location matter when selecting a solid state provider? So yes, we take geographical proximity into account. No, we take a global approach. Or neutral, geographical proximity is not a factor. So please answer that, whether geography or proximity matters to you when you're selecting a solid state provider. Oh, that's interesting. We have 22% saying yes, we take geographical proximity into account, 22% no, we take a global approach, and 56% say they're neutral on geographical proximity. Edwin?
Mm-hmm. Yes, well, as the experiments will be done in our time zone, of course, and but when we talk about the results, we have a project manager or project leader who adjusts to the time zone of the customer, and that should never be a problem. Of course, when it is close by and you can easily check what is going on, you can discuss face-to-face, that is better. But you also have a choice of indeed the best expertise or the highest quality, then you go for that one.
Right. And Edwin, we do have a question from the audience here. When do you conduct a polymorph screen? At pre-nomination, as part of the scale-up work in phase 1, or before phase 2?
Well, of course, as early as possible. But when it is really a polymer screen, when you look at the different forms which are there, you need to have to make the choice of your molecule already. So in this case, most of the polymorph selections, the polymorph studies, are done just before going into the tox study, so before phase 1.
Okay. And our final question, Edwin, is have you encountered isolation problems around intermediates, such as very slow filtration?
Yes, we had several of these, and I just talked about one which was a very slow filtration, but drying because water was trapped into cavities was also one of these. Each filtration problem has a different cause. It can be conditions of your crystallization, but it can also be the temperature at which you try to isolate it. And when you go down in temperature, you attract more of your impurities and you go to a less crystalline form there. So each is approached differently. And that is very much, well, the difficulty of the solid state, that none of the projects are actually identical.
Right, they're unique then. Okay. Thank you very much. That's all the time we have for today. I would like to thank everyone for attending today's webinar. And don't worry if you missed part of the presentation. Simply visit www.excellience.com, where you can download any of our archived scientific seminars. If you want to share this presentation with your colleagues, simply note their name and email address in the post-webinar survey, and we will then send them a link to the archived presentation. Please join me in thanking our speaker for today, Dr. Edwin Aret, who is a solid-state scientist at Cambridge Major Laboratories, Inc. You will notice that a quick post-event survey will pop up on your screen at the end of today's seminar. Please do fill out this survey because we would really appreciate your feedback as well as your suggestions for future webinar topics. Thank you all for your time. We hope you found this webinar informative. Have a great day.